Good evening, ma'am.
Good evening, sir.
Good
This is
evening,
Mahindra. Yeah,
sir.
I will make you host. Yes, sir.
Okay, sir. Thank you. Thank you, sir.
Yeah.
Sir.
Sure.
Thank you, sir.
Yeah.
So let's start, sir.
Yes, I think, yeah, you can start. Yeah.
Yeah. Okay. Thank you, sir. Good evening, my dear students. Today we are going to discuss about the remaining part of the biotechnology. So already we discussed what are the types of biotechnology. So you know about the red biotechnology, white biotechnology, and then green biotechnology and blue biotechnology. Okay, each one it has a specific properties and analysis. Okay, already we saw it that things also. And then so genetic engineering. So what is the meaning of genetic engineering and how it will be proceed? Okay, the technique. That things also already last class what we discussed, you know, isn't it? So today what we are going to discuss, you know, the remaining part of the cloning and other related headings. Okay, see that one. Okay, last class what we discussed.
Sir, can you please record this session? Click on the record button.
Okay,
yeah,
yes. Thank you.
Yeah, thank you.
Can you, sir? Is it
yes.
okay, sir?
Yes, yeah, please.
Yeah. Now, okay, sir, isn't it, sir?
Yeah.
Okay, thank you, sir. Thank you. Okay, today what we are going to discuss, you know, the gene transfer method. So you know about what is meant by gene. Before that, we know how to gene are isolated from the other organisms. You know, but there are so many methodologies, the rupturing of the cell wall, and also that other cell-bound organelles also we collect. After that, it undergoes the process. What is that? A centrifugation process. After that, it undergoes the amplification. Finally, what happened? You know, if a purification is also we have to do. So after that, what happened? You know, so what are the methods that were used for the gene transfer from one organism to another one? See here, the first you should note it down. So, DNA is the genetic material. Already you know about it is transfer the characteristics of the one generation to another generation. It's otherwise called as DNA inheritance process. Okay, already you know about it. It's a high molecular weight genetic material present in the cell, isn't it? Here, the DNA is a hydrophobic molecules because it is not easily dissolved with the water molecules. That's why it is called as water hatting. Okay, and cannot pass through the cell membrane. It cannot easily penetrate or pass through the cell membrane because it is made up of what is that, you know, lipoprotein. The cell wall is depends upon the plant or animal. The cell wall is made up of what is it? Each one it has a specific component. For example, plant cell is made up of cellulose. Is one of the polysaccharide, isn't it? Likewise, the animal cells also. The animal cell also is made up of what is that, you know, lipoprotein. Okay, so the DNA molecule it's not comes out through the cell membrane. So then what will happen? The gene transfer techniques also grouped into two main categories. How the genes are transferred, what are the techniques that were used, what are the equipments, otherwise called as what are the molecules used for the transformation of the gene from the one organism to another one. How it will be, it majorly divided into two types. One is called as vectorless or direct DNA transfer. The direct DNA transfer, otherwise called as vectorless. In between, there is a vector, it's nothing but the mediator. Okay, they were used for the gene transformation method. The first one, what is that? Vectorless or direct DNA transfer. Directly means what is the isolation of the DNA molecule, which one we are going to introduce into the host cell. You have to find out, and you have to use the another one, gene gun. That is very, very important one for loading the DNA molecule. So then it will be targeted to the exact cell. So that is called as direct DNA transfer or a vectorless transformation, gene transformation. So another one is called as vector mediated transfer. So vector mediated gene transfer, nothing but there is a mediator between the host cell as well as the foreign cells also. So these are the very, very important things. So now we will see one by one. Okay. Okay. See that one.
Good evening, sir.
Good evening, Ajay. Okay. Okay. See that one. The first one, the vectorless or direct DNA transfer. See that one. It has been further subdivided into two categories. So according to the vectorless means there is no any vector in between the gene transformations. So this is also further divided into two types. One is called as physical gene transformation. Another one is called chemicals. So there are two types have been reported. Okay. See one by the physical gene transfer method. Okay. How it will be. So already we discussed this is very, very important method for the gene transformation. Okay. One is called as electroporation. Electroporation. The electroporation is the process where the electric impulses of high field strength are used to reversible permeabilize cell membrane. Okay, facilitate to uptake of large molecule including DNA. So what is meant by electroporation? Electroporation means with the help of the electric. Poration means nothing but we are going to make a small hole. Then only the molecules it may be penetrated into outside to inside with the help of the instruments like this. See that one? In electroporation, the external electric field is applied to the, which change the electrical conductivity and the permeability of the cell membrane. Thus, the exogenous molecules, exogenous means outside the molecules, found in the medium or taken up. This is also very, very important. The electroporation is nothing but the method used for the, what is that? Uptake of large molecule, including DNA. Fully, we have to absorb the molecule. Okay, so including the other cell-bound organelles also. As a result, what happened? See that one? See, this is the process. So before pulses, this is the cell membrane. Just imagine. The cell membrane is also surrounded by, which one cytoplasm. Inside there is a cytoplasm is there. And outside also there is a, what is that? Other molecules also present in it. It has its own specific charges, whether positive charge or negative charges. Okay, when you are during the electric field, when you are giving the electron, a conductivity, what will happen? The positive charge are arranged because electron we are giving, isn't it? So it outside that is a minus charge is the corresponding positive charge is also what happen? Arranged in a line inside the cell wall. So there is a pore, there is a small gap in between the cell membrane. The molecule can be easily inserted from the outside to inside. Okay, so the last one, see that one? There is a molecule there, in and out. After that, what happened? Completely heal it. Okay, this process is called as electroporation. Last class we have discussed. Okay, so this is the method. And then second one is called as very, very important method, that is called as particle bombardment, otherwise called as microprojectile or biolistic method. So this is also very, very important. Sometimes they may ask the question, what is by particle bombardment is otherwise called as what is that? Microprojectile, and also biolistic method, otherwise called as biolistic method, also called as what is that? Like that they were asked the question in NEET point of view. Okay. Sometimes they were asked the questions, these two methods where it will be used. Okay, in the DNA fingerprinting or isolation of DNA, cloning or some other else. Like that they may ask the question. Okay, see that one. The technique of the particle bombardment, also known as the biolistics, microprojectile bombardment, particles acceleration or gene gun, etc. These are all are that, you know, there is a method to introduce the what is that gene into the target tissues. So it's the most versatile and effective way. These are the methods mostly highly effective way to introduce the gene transformation method. It employs a gunpowder driven device and helium driven particle guns. These are all are very, very important. Okay, just like a similar to that of our gun only, but it is specifically made for the gene transformation. See that one. The DNA bearing is tungsten or gold particles, 1:3 in diameter. Sometimes they may ask the question, the gene gun. Okay, genetic transformation gun is also made up of which metals? Tungstens and gold particles. Okay, the size will be how it will be, 1 to 3 diameter, referred to as a microprojectile, is accelerated into living plant cells. I will show you how it will be. Okay, yesterday we saw it, the analogy is in the classroom. So we saw it. So, and then next one is DNA coated onto the microscopic pellets. The microscopic pellets also, DNA coated onto microscopic pellets. It's also make it as a DNA, is make it as a pellet form. It's literally shot into the targets. Then only it's why it is make it as a pellet form, then only it's pellet is nothing but the bullet. Okay, the gun is loaded with the bullet in normal function. In this gene transformation, that gun is what is that made with which load with the pellets. The pellets contain what is that DNA molecule. Okay, so then only the DNA of the foreign DNA, it reaches its target site. See that one. Next one, particle bombardment is used to deliver drugs, fluorescent, dyeing, antigen, protein, etc. Whatever it may be. How it will be bombardment? Bombardment is nothing but what is that? So we are introducing into the cell only, within the cell only. How it will be? With the help of the gene gun only. So this is very, very important, my dear students. See that next slide, you can understand the concept. Okay, so this is the instrument of gene gun. So already it loaded, which one pellets. Okay, the pellets contain which one DNA molecules. This is the sample plants. It has a own specific diameter also, and also we are shot the gun. So during the process, so this is the method they were used for the gene transformation from the plant to animal, animal to plant, whatever it may be. Here, what happened? The isolation of the DNA molecule. Where will you get it? We do not know, isn't it? It may be any plant sources or animal sources. After loading in the form of pellets, it can be transferred to the gene gun to plant. So this is the gene gun. So go to the next one. See the different types is there. Gene guns. Okay, helios and by the bioradar is used to transfer cell in culture and plant leaves. This is called a cell culture. See that one. This is the instrument. Already know what gene gun is there. Okay, there is a handle. This is here the loading process also there. Okay, so there are so many parts is also there. We have to properly use it. So then only we can easily transfer the gene molecule from the one place to another place, or one organism to another one. In this category, in this first one, just you are seeing that first one, it is nothing but the cell culture. Here, the cell culture, it contains the microorganism inside it. So here what happened, there is a DNA molecule loaded. The DNA molecule transferred to the cell culture through the gene gun. Okay. Another one already we saw it. This is also another one. It is for plant leaves. The gene transformation takes place from gene gun to where the plant. Here, cell culture. Okay, here, source. What are the next one? So this is the structure of, okay, the gene gun barrel. So barrel is nothing but a small protrude like structure. That means the outlet. Seriya? Small outlet is made up of, what is that, tungsten. Normal gun barrel is also made up of, what is that? Iron, isn't it? But in this category, the gene gun barrel is made up of which one, tungsten or gold plate. So here there is a small, what is that? Plastic disc, and with the DNA coated gold particles. Here the particles also loaded. Then the disc stopped by the screen. There is a small screen also in front of the, which one, the plastic disc. Okay. So now what happened, the force when we are giving the gene, okay, when we are sort, what happened, the loaded DNA molecule, it's also transferred through this way. Finally, it reaches the plant target cells here. Okay. TNA, plastic coated, the gold particles, the gold particles, when it reaches the target cell, it's also incorporated with the cell. So this is the process. Once again, repeat for you. Gene gun barrel, this is the gene gun barrel. Here the loaded molecule is there. It's nothing but the DNA molecules from the foreign particle. After that, what happened? The gene barrel, tip of the gene barrel, what happened? So there is a plastic disc is there with coated with the DNA gold particle is there here. After that, a few inches, what happened? There is a disc trapped by the screen also present in it. Okay. So when we sort the molecule to the target cell, what happened? It pass on this way. Finally, it reaches the disc and also the culture plate also, target plant cells also. These are the techniques. Okay. See the next one. The particles coated with the foreign gene releases the foreign genes when they enter into the target cell and integrates into the chromosomal DNA. Integrates means both host DNA as well as the target molecules. Which one, inside the cell, inside the plant cell or inside the animal cell. Okay. The gun is already, but it is nothing but the foreign particle. Which one is we are going to introduce it? So selectively, okay, it may be highly resistant to the plant or animal, depends upon the superior characteristics. That is also loaded, isn't it? It is also reached to the DNA. As a result, what will happen in the both the genes are integrates within it. So as a result, what happens? The multiplication takes place inside it. So the two chromosome DNA molecule also culture inside it. Okay. As a result. Yes. Okay. Next one is micro injection is also another type of the method. It is the injection of the DNA solutions. It is the injections. Injection means already what are the things we need? Dhanalakshmi, injection we need. Hello. What about Ajay? Okay.
So,
yeah, Ajay, what do you need? Injection. When you are giving the injection, what are the essential things? Needle.
so needle, sir.
Okay. Then coast is also very, very essential, isn't it? Target cell.
Yes, sir.
Then what about the antibiotics? Okay. So after the loading, we are introducing into the. Okay. So just see here. So it is the injection of the DNA solution by micropipette into cells. The glass microneedles are used for the transfer of micromolecules. This is very, very important, my dear. Glass microneedles, okay, are used for the transfer of the micromolecule into the cytoplasm or the nucleus of the recipient cell of the protoplasm. Okay, see here how it will be. Already saw it, these things. So see one. Yesterday we saw it, this one. So this is considered a plant, isn't it? A plant cell. Take an example. This is considered this is considered a plant, isn't it? Just imagine. Okay. Okay. This one is plant. When you're taking any section of this part, okay, when you're going to take any section, so you'll get the structure will be like this. Okay, inner what happened? The cell is like this, isn't it? Can put hexagonal structure. Okay, this is considered a cell. Center what happened, already you know about the, so one of the nucleus. Nucleus is the each and every cell, there is a nucleus. Okay, from this one, we are going to separate the nucleus. How it will be, you know about the, we have to isolate the nucleus. The nucleus inside what happened, there is a DNA molecule. So DNA. So DNA molecule is there. The DNA contain what is that? Chromosome is there already in our report. Okay. So not only this one. Suppose take an example. So this is plant, isn't it? Plant sources. This is plant sources. This is considered as a plant sources, isn't it? Suppose if it is animals, take an examples of animal source. Okay. Animals means just imagine. Take an example, say any one of the prokaryote. Prokaryote means bacteria, isn't it? Bacteria inside there is an extra chromosomal body present in it. That extra chromosomal body is called as, what is it, That is called as, what is that? This one is considered as a plasmids. Okay. So plasmids. Okay. The plasmids is called as extra chromosomal body. Okay. It is present in the bacteria because both are having, which one is the plant source, this one is called as animal source. The plant cell is made up of cellulose. Yesterday we saw it, cellulose. Okay. So very, very important, my dear. Cellulose. Cell wall is made up of cellulose. Here, what happened? The animal cell wall is made up of lipoprotein, particularly in our board, lipoprotein or lipoprotein. Okay, protein. Otherwise called as phospholipids. Okay, TNIIT. Okay, so lipoprotein. So the cell wall is made up of lipoprotein. How it will be degraded? So how it will be lysis? How will you lyse the cell wall? And also we can isolate the DNA molecule from the cell. How it will know? Because each one it has a specific enzymes. Okay, so that is very, very important. Sometimes what happens? There is a cell is there. So just imagine this is considered a cell. So whether it may be a plant cell or animal cell, whatever it may be, the center there is a nucleus is there already in our board. This is considered a cytoplasm. This part is considered a, what is that? Cytoplasm, isn't it? This one is cytoplasm. Cytoplasm is there. This one is called as nucleus is there. Not only the nucleus, and also along with it, that there are so many cell-bound organelles is also there. This is called as endoplasmic reticulum, Golgi apparatus. Golgi apparatus present here. Okay. And also mitochondria already in our board. This is considered as a mitochondria, and ribosome is also present in it. A different structure I have to drawn here. So you all are found in varying in the cytoplasm. The center there is a nucleus. All the activities controlled by the nucleus only. Okay. So suppose if I introduce any one of the target DNA molecules, means what is that? So we have to lyse this component. This cell wall we have to lyse with the help of a specific enzyme. Suppose the plant cell wall is made up of cellulose, means we were used which one? Cellulase enzyme. Cellulase is the enzyme. The cellulase easily act upon the cellulose. So as a result, what happened? The cell wall easily broken. Lipase, likewise, the lipid molecules, it may be easily broken. Likewise, what happened? We can easily rupture the cell wall. Okay. Then only we will get the nucleus molecules. Okay. Okay. See that one. So stop sharing. Okay. See that one, my dear. Sorry.
Sir, your screen is not seen, sir.
I will show you. But now, are you seeing?
Yes, sir.
Yeah, sorry. Okay, my dear. So last class we discussed this part. Okay. So today what we are going to do. So next things is called as what is that? Gene gun method also we saw it. And the next one is called as what is that? So micro injections. So see here. So the glass micro needles also very, very important. Then only the micro needles used for the transformation of the molecule from the outside to inside. There may be a cytoplasm. The introduction may be where into exocytosis. That means outside to inside cytoplasm. And sometimes it may be reaches the nucleus, and also sometimes it may be a hepatoplasm. So during this process, what happens? The target cell, it will not move. That is very, very important, my dear. Target cell, it will not move. See that recipient cell must be kept immobilized in nature. Then only the target, it will be reached very easily. When you are sort, what happen? The DNA molecule will be reaches the exact target cell. The cell is held against the solid support or holding pipette, and micro needle containing the desired DNA is inserted into the cell. This is very, very important. The micro pipette needles or whatever it may be contain what is the name? Desired DNA molecule. That is already we discussed yesterday. Okay. Then the delivery of foreign DNA is done under the powerful microscope, micro manipulator. Okay. We cannot able to see that naked eye. So that time what we have to do, you have to use a powerful microscope. Then only it will be reached whether the target cell or not. The things we can see. See, this is the holding pipette. This is the cell. Okay, this is the host cells. So the center there is a nucleus is already know the in between the nucleus and the outermost layer is called a cytoplasm. Already we know these things also. So this is the micro pipette, otherwise called as micro injection or needle, whatever it may be. This is the needle. Already know it look like a syringe. Inside what happened? This portion, what is it? The foreign DNA molecules. Okay. So you have to introduce inside exactly target cell. Means what happened? The host cell, it will not move here or there. Okay. So the holding of the host pipette, this one, the cell is completely held by the holding pipette. Then only the DNA molecule exactly it will reach the target cell. So this is very, very important. So go to the next one. It will be better for your understanding. This is the male pronucleus. This is the nucleus. Simply, this is called as holding plate. Pipette. Okay. See the needle is that. This is the needle already in our about loaded. Okay. So the needle is how it will be. See. So the needle is exactly target to the cells. As a result, what happens? Once it reaches, see that one. This is the correct way. So this is when you are introducing the target cell. It will reach us, introduce the foreign DNA into the host cell. So finally, what happened? It comes out. So successful injection with swollen pronucleus. So as a result, what happened? How it will be identified? The foreign DNA molecule, it reaches the host DNA means what is that? Swollen pronucleus. Pronucleus and structure will be how it will be? Swelling. Okay. So these things also we can easily see under the powerful microscope only. We cannot able to see the naked eye. That is very, very important. Okay. See that one. So this is also another part. This is the holding plate already in our report. This is the cell, okay, holded by the holding pipette. So this is the target cell, otherwise called as nucleus. So here loaded, this syringe is the syringe is inserted, and also loading of the molecules. So as a result, what happened? The size of the pronucleus will be very large or bulbs. That is very, very important, my dear. After reaching what happened, inside the recombination also takes place automatically. That is very, very important, my dear. See that one. Next method is called as liposomes, mediated transformation. Liposomes or lipofixin. Liposomes are artificial spherical lipid vesicles surrounded by a synthetic membrane of the phospholipids, otherwise called as compound. Phospholipids and phosphorus combined with the molecules of lipid, that is called as phospholipids. So you have to remind in this part only. Okay, how it will be? So listen here. Liposome are artificial spherical lipid vesicle surrounded by a synthetic membrane. Okay, this is the artificially made. Okay, so this is made up of which one? Phospholipids component that imitate the structure of biological membranes. It's exactly similar to that of the which one? Biological membrane, because The biological cell, it contains what is there? The center there is a nucleus, the outside there is a covering is there. That covering is also made up of which one? Phospholipids, already told you. Okay. Likewise, the manually they made it. So then this can be introduced to fuse with the protoplast using, this can be, which one? This component, artificial spherical lipid, otherwise called as lipofixins, is what is that? Can be induced to the fuse with the protoplast with the help of the one of the connective substances. Don't forget, my dear, this is very, very, very important. PEG, polyethylene glycol, mostly used for the protoplasmic fusion. Polyethylene glycol, protoplasmic fusion. Okay, then what happened here? Addition of this chemical substances, what happened here? The two components, which one is the artificial lipid component as well as the protoplasm, it easily joined together, adhere together. Okay, DNA to transfer is packaged into the liposome in vitro and transferred to the target tissues. Okay, so DNA enter into the protoplast due to the endocytosis of the liposomes. Okay, so once see that, once again repeat for you how it will be. See that one. See, protoplasm, yesterday we saw it, there is a two plant, take an examples, any plant, take an examples of the any plant. Okay, take an examples. Here also another one. Okay, this is the here B. Okay, like that, imagine. So this one is considered as a plant. Okay, this one is considered as a B plant. Okay, just imagine this is A, so this is B. The both are, they are having the cell, isn't it? Each cell, it has a specific structure. Okay, a hexagonal structure. Okay, inside what happened now, there is a protoplasm is there. Yesterday we saw it, that things also. Here there is a protoplasm is there. Okay, a protoplasm is violet and another protoplasm we can use it as a orange color. Okay, this is the A. Okay, this one is B protoplasm. The protoplasm both are, how it will be united means you have to rupture the cell wall. So as a result, what happened? Rupture the cell wall means we are using different type of enzymes, particularly the plant cell is made up of cellulose. So we are using the cellulase enzyme, cellulase cellulase. Okay, the enzyme. Simply we were use the cellulase enzyme. So as a result, what happened? The inner side there is a protoplasm is the protoplasm is comes out. Likewise this. Okay, protoplasm it will be isolated. Likewise, this here also. Here what happened? This A molecules also comes out. Here we have to find out. Which characters, which gene is responsible for the restriction genes. For example, this one is considered as a restriction genes. Any one of that. Okay, any one of the restriction genes. For example, tetracycline. Suppose this is considered as a resistance, rough for ampicillin or penicillin. So take an examples, ampicillin, AMP, this particular genes only. So now we have to remove this part. So this is the desirable character, or this one is whatever it may. Which one is the superior character? Okay, we may take this one. It means so you have to remove this part. This is gene is a interested one. This gene is a interested one, isn't it? This genes we interested one means we have to cut the gene from the organism, isn't it? So we are using the which one restriction endonucleases. As a result, what happened? You will get the gene like this, isn't it? You will get the gene like this. See, this is the part. So there is a desired gene like this. Okay, this is our target gene. This gene is introduced into the host cell of the another plant. Take an example. This is the here also you have to make a cut and this part is completely removed. So as a result, This part will be like this. Okay, this part will be like this. Okay, so this is the removal part. This is the removal part. This one is we do not know, means we have to cut it and remove it. So this is removed. Okay, this one is removed. Okay, removed, and then we add this part because the target cell TET. This is the resistance. Interested gene is that this interested gene is introduced to this host cell in this part instead of the ampicillin. Instead of the ampicillin, we are introducing the TET. So as a result, what happened? You know, the complete structure will be like this. Complete structure will be like this. How it will be? See that one. Structure is, so this is the structure, entire structure. So look like this one. Okay, this is the exact structure of the hybrid. How it will be? See that one. So this one, 11. So this one. Okay, so this one is a gene, isn't it? This is the A gene. This one is what is that? This one is A. Sorry, this one is a. Sorry, this one is not A gene. Okay, so this one is a B gene, isn't it? So this one is a gene. Is it correct or not? This is the A gene, isn't it? This one, this part is A gene. But in this part, we removed, you know, we are introducing here what happened oppositely. Okay, so this part is introduced here. So likewise, we may use this part to here. How it will be? See that one. This is one part, and another is also we can make it. Two types of hybridization also we can do it in this part. Okay, so in this part we may mix with the. So here imagine this is the A gene incorporate with the B gene. Here A gene is a violet color and B gene is a orange color. So here the this orange color indicated AMP, isn't it? Ampicillin. Ampicillin. Here the violet color indicating tetracycline, isn't it? So after that, we can these two hybridized molecule and incorporated molecules introduce into the host cell. That is very, very important. It introduce inside the host cell. How it will be, you know? So first of all, you should note it down. We have to select the two different plant cells, and from this one, we have to isolate the plasmids. Okay, and after that, we have to identify the plasmids, which is interested genes. That interested gene also isolated. After that, it incorporated with the specific genes of the another one. Okay, A with B, here the A with B. So after that, this completion of this one, during this combination, so what happened in the fusion of the protoplasmic fusion, what we are using, you know, one of the substance, which substance they were used, you know very about, that is considered as that is named as which one, you know, PEG, that is called as polyethylene glycol. So this combination, the adhesion process or connective process, what we are using, you know, one of the chemical substances name is called as PEG, that is called as polyethylene glycol when we are using here. After this one, we have to introduce the, okay, so we have to introduce after the completion of this process, we are isolating the DNA molecule from the different sources. We have to introduce in the where? In the vector, otherwise called as, that is called as E. coli. Take an example. So this is considered as E. coli. Take an example. This is the E. coli bacteria. Okay. Which one we are selecting? You know, suppose if you are selecting this part, this one I want to amplify more than 100 times or 1000 times or whatever it may be. This part I want means it can be introduced into the host cell. This is considered as a host. Maximum the host cell it will be which one, you know? Host cell is the, this is the host. Maximum they were used E. coli. So E. coli otherwise called as what is the Escherichia coli. Okay, now we are introducing this one with the help of the micropipette or injection or gene gun or whatever it may be. How it will be inside? See that one. Inside how it will be there. So the structure will be like this. Okay, just imagine this is the inner part of the introduced gene. This is considered as a foreign gene. Okay, this part. This is the A part. This is the B part of the DNA molecule. So both are combined together, isn't it? Now this is undergoes the replication process, replications inside the host cell, because they need the host, then only the replication process is completed. Otherwise, it will not complete. So inside what happen, the replication process occur. As a result, these combined genes, A and B genes, combined genes, you will get it. How many types is there? See that entire structure will be like, look like this one. Okay, see this one. There are so many copy of. Okay, so many copy, it will be produced. Both the gene present in it. Both the gene present in it. So this is the. Okay, so likewise, you will get it different copies, number of copies it may be. We cannot be able to calculate how much you need. These are all are, what is that? Cloned molecules of both A plus B genes. Okay. Now what happened, which one it has the resistance of tetracyclines? Okay, tetracycline here. So all the genes, they are having the resistance against the tetracycline. So the microorganism also fight against tetracyclines. So this is the very, very important. This is the methodology we are using in clinically. Are you understand, Ajay? Ajay, do you understand?
Yes, sir.
Okay. What about Dhanalakshmi?
Yes, sir.
Okay. See this one. See that one. Okay. So we'll pass on to the next one is ultrasonication or ultrasound mediated DNA transformation. See that heading, ultrasonication or ultrasound mediated DNA transformation. The ultrasound is used for the stimulating uptake of foreign DNA. Ultrasound is used for the stimulating of uptaking of foreign DNA by plant protoplasm and leaf segment of tobacco. This is very, very important. This is the okay. And then the UV laser micro beam also used for the, what is that? Foreign DNA molecule. It creates a transient pose in membrane for DNA uptake. Suppose if you want to take which one uptake of the DNA molecule from the cell, means we are using which one, ultraviolet laser microbeam. That's why the heading is also ultrasound mediated DNA transformation. Then another one is called as calcium chloride, CaCl2, with DNA precipitations. Why? When you are treating with the calcium ions, okay, what happen, so it is automatically the precipitation takes place. How it will be, calcium chloride is addition with the DNA molecules. What happen, the treating of cells with the calcium ions increases the membrane permeability of the DNA uptake. Okay, so as a result, the calcium usually bind with the cell membrane. It lose their ability. So the DNA molecule uptake, it will be very easy. That is the point here. Okay, so the ultrasonic or ultrasound mediated DNA transformation, here they were used a UV laser microbeams for the DNA uptake. At the same time, they were used the chemical substances such as a calcium chloride, DNA precipitation method, like that they told, because the DNA precipitated, when you are adding the calcium chloride, what happened? The cell membrane increase the permeability of the DNA molecule from inside to outside. Okay, so this is very, very important. See that one. This is the structure. This is any one of the bacteria. Take an example here. Also, the Bacillus bacteria has given, and also we are adding calcium, otherwise called as calcium chloride. As a result, what happened? See here, these are all are okay. It can be easily penetrate. See that one. The molecules completely give a, what is that? Penetration. There is a pore making process because the cell wall is made up of phospholipids usually bind with the calcium molecules As a result, what happened? There is they make a pore-like structures. As a result, what happened? It will be very easy. The molecule, it will be easily penetrated inside. That is also important. This is the plasmids. It can be easily moved to here to inside. See that inside? Okay, this is the structure. So another molecule, when you are giving the heat soak, during this process, we have to give a heat soak also. That time, what happened? The rupture of the cell wall like this. Okay, the molecule is also enter inside and also completely closed. Surviving bacteria has taken up the plasmids. Those who are surviving, such type of bacteria easily absorb the plasmid molecule. It can be easily survive. Those who are not surviving, it may be completely destroy the molecules. It releases the molecule into the cytoplasm, and again, that molecules also select the another one. Once again, repeat for you. This is the bacteria. Bacteria structure inside there is a chromosome is there. Okay, when you are giving the calcium chloride molecule, what happened? The permeability of the cell membrane, it will be very easy. So as a result, what happened? The plasmid will enter inside during the heat soak. So those who are not survival, those who are not surviving in this methodology, in this field, that is calcium chloride containing field, what happened? All are destroyed. Those who are surviving, that can be, what is happening? Easily taken up the plasmid molecules. This is the important things. Okay, here the transformation takes place with the help of calcium chloride only. Plasmids, we cannot able to introduce any gene gun, anything, we will not use it here. We used only calcium chloride medium. Okay, and also we are giving it the heat. During these two combination, what happened? The cell wall easily penetrate the plasmid inside the bacteria. So which one is receiving the bacteria? What comes? It is considered as a plus. Okay, H plus, otherwise called as bacteria plus. Suppose those who are not having the bacteria, they are all considered known as minus category. Okay, so now what happened? These are all are minus, but in this category, this one is considered as a positive category. Otherwise called as highly frequency factor. Sometimes they call HFR factor. And also another one is called as fertility factor. Sometimes they may told, okay, which one bearing the chlorophyll, plasmid molecule, such type of bacteria is called as, what is that? Fertility positive bacteria. Okay, it is not related to the gram positive, gram negative. Just imagine the extra chromosomal body of the plasmid present inside the bacteria. It is considered as a male or fertile factor. Suppose those who are absent, such type of bacteria is called as negative bacteria. Okay, I mean fertility factor negative, not the gram negative, gram positive. Okay, just imagine. And heat soak, how much it will be? How many degrees Celsius we have to give? How many seconds we have to give? See here, 90 seconds. It is equal to 1.5 minutes. Competent cells are given a brief heat soak, 42 degrees Celsius for 90 seconds to relax the cell wall and high temperature stress causes up to regulation. The reactor responsible for the DNA recombinant and repair. So when you are giving these things, what happened? It automatically the plasmid molecules enter inside that. How it will be enter already but there is a small opening is there outside the cell membrane. It completely healed. It also come to the original position of the structure. So that was done by these two components. What is that? Calcium chloride, as well as heat we are giving. Heat, how many minutes we have to give? 42 degrees Celsius for 90 seconds only, 1.5 minutes only. That time what happened? Completely receive that. What is that? Heat. And stress causes up to the regulation of the reactor responsible for the DNA recombinant and repair. So as a result, what happened? Completely the DNA molecule, the penetrated inside, and as well as the cell membrane is also recover once again. Reformation also takes place. This is also an important thing. And the next one is called as chemical gene transfer method. This is also another important idea. This involves the plasma membrane destabilizing and also precipitating agent. The agent means nothing but the chemical substances. What are the destabilizing? Destabilizing, it can be easily break. Precipitating means already it's settled down. See that protoplasm are mainly used being incubated with the DNA and buffers containing PEG. So always the protoplasm, it will not store. Suppose after the isolation of the plant cell or animal cell, the protoplasm, where will you store it? No, with the help of the one of the buffer system. Yesterday I asked you what is meant by buffer, sodium todecyl sulfate, where will you use? Okay, that things also already we discussed, isn't it? Polyethylene glycol here. Polyethylene glycol is nothing but the one of the buffer solution used for the storage purpose of the protoplast. We can isolate the protoplast. The protoplast is nothing but there is no any cell wall. A small minute layer only is there. Inside there is a genetic material present in it. Okay, when you are joined together two protoplasts, you will get the new variety. Okay, so that is a important one. See that containing PEG, polyethylene glycol, and polyl-ornithine, polyvinyl alcohol, or divalent ion calcium chloride. These are all the chemicals used for the gene transformation method in gene cloning. Don't forget, my dear. What are the chemicals that were used? This is very, very important. PEG, don't forget, PEG, polyethylene glycol. Mostly they were used for the, what is the protoplasmic fusion. Okay. When you are using the PEG, what happens? The protoplasm is easily fused together. As a result, you will get the single protoplasms. A strain as well as the B strains, isolation with the help of the PEG, it will be united together. Okay, but here the buffer using system. Buffer means what is the, when we are adding any one of the chemical substances, the pH, it will not change. Such type of solution is called as what is the, buffer. Okay, not only the PEG, other components also given here: polyl-ornithine, polyvinyl alcohol, or divalent ion calcium chloride. Sometimes they are asked which one of the divalent chemicals used for the gene transfer method. Calcium chloride. Okay, this one is very, very important. Polyvinyl alcohol. So 4 is very, very important. Particularly these two are very, very important because the PEG is mostly used in the protoplasmic fusions. Then we'll pass on to the next one is a question part. Here already we discussed who coined the term biotechnology for the first time. Ajay and also Dhanalakshmi. Who coined the term biotechnology for the first time means there are the 4 options is the Hamilton Smith, Carl Erke, Northens 1 and 2. Already we know Carl Erke. So many times I told you Carl. This is direct questions. This one is a direct question. Carl Erke. So other options also you have to note. Hamilton Smith, which one he discovered or introduced, which concept he stated. Northens, what is that? What will be the process? That things also you should note it down. Then only we may find out the correct answers. We can find out the correct answer. Next one. The biotechnology has become an important tool in crop improvement. Okay, yes. Human health care, yes. Chemical industries, yes. So these 3 importance, okay, tools: crop improvement process, human health care, and chemical industries also. So that all of the above is the correct answers. Okay. When the questions, when you are reading read the NEET questions, what happened, there are 4 options is the right. Particularly 2 options is related to the subject, a question. Remain 2 is not related to the questions. You have to find out that one first. After that, the above that related one, you have to find the exact answer. That is very, very important. Okay, so see that one. Next one. The traditional biotechnology include the process that are based on which one? Genetic engineering, traditional biotechnology, modification of gene chemistry, natural capability of the microbes, both 1 and 2. The 1 and 2 is not involving traditional. It is not a traditional biotechnology. Natural capabilities, what they are having of microbes. This is the correct one based on it. Because naturally, what are the capability? What are the DNA molecule or protein molecule, carbohydrate, amino acids, enzymes? What are the component present in the microorganism, otherwise the animals also. Okay. It will not related to the first 2. Okay. This is important. And the industrial fermentation is still in use for the production of Which one? Industrial fermentation. Curd, you know about, yes, curd is also using. Preservatives, preservative used for the curd process, making process. Enzymes. What they're asking, industrial fermentation is still in use for the production of enzymes only. Traditional. Okay, so industrial fermentation is still used for the production of enzymes only. Curd in previous category, what in ancient ages, what is that? The curd as well as the preservative is not used in industrial purpose. Okay, this naturally we made, isn't it? But this is also the enzymes only we made in the industrial fermentation, still in use for the production of enzymes only. Okay, and then the modern biotechnology depends upon the fermentation, change in chemistry of genetic material, and plant breeding, both 1 and 2. Okay, both 1 and 2 is not involved because fermentation is nothing but a simple process, a process. It is not comes under the, it is not as a biotechnology. Okay, take an example is nothing but the chemical process. Take an example, milk is also undergoes the fermentation process. As a result, you will get it the, what is the 2 process there. One is called as lactic acid. With the help of the enzyme lactase by the microorganism is called as, what is the lactobacillus, isn't it? And another category, the fermentation is of the ethyl alcohol. You know about the sugar. Molasses is also converted into ethyl alcohol and carbon dioxide with the help of the enzyme is called as invertase and sucrase. This process. This is not comes under the, it is biotechnology. Okay. Change in chemistry of genetic material, because the biotechnology means it's also one of the genetic material changes only. Chemical structures also changed. Plant breeding. Plant breeding is nothing but the one of the physical methodology. One, these two are physical even. This one is the, what is that? Biotechnology. Technically, we were used. That is very, very important. So the change in the chemistry of genetic material is the correct answer. Others two are not coming this part. Okay. So that's all about my dear students. Today this class is over, and also this year also this class is over, my dear. I will summarize slightly for what are the headings only we will discuss. What are the things we discuss, See, first of all, you should note it down. The physical gene transfer method, we are used the electroporation. And second one is, what is it, So the particles bombardment, a microprojectile or biolistic method. Here, how the gene gun is made up of which one? What is the particle size? That is very important one. How it will be loaded? That is very, very important. Pellet form. The DNA is also loaded in the form of pellets. Okay, this is also very, very important idea. This is the gene structures already about how it will be introduced in the tissue culture as well as the and cell culture also, whether it be animal or plant. How it will be the gene gun is loaded. See that this is the barrel already but there is a plastic disc is also coated with the golden particles. Along with the which one there is a disc stabbed by the screen also there. Here there is a molecule is there. It is when it sort, what happened automatically it reaches the target cell. So this is very, very important. After that we can analyze with the help of the powerful microscope what happened inside. Okay, so this is very, very important, my dear students. And then another one is called as micro injection. In this part, micro means very, very minute product, micro liter. Okay, we loaded into the micro needle, and also it also reaches the target cell and also transferred to the target cell, whether it may be a cytoplasm or nucleus or protoplasm. These are the 3 parts only. Okay, the recipient cell must be kept in immobilized. Okay, the target cell, it will not move here and there. Okay, so that is very, very important one. So we are using specialized structure is called as what, holding. Okay, so then only it will be reaches the exactly. The delivery of foreign DNA is done under the powerful microscope, micro manipulator, otherwise called as micro manipulator. No, don't forget, my dear, this is also very, very important, my dear. And also this is the structure already but the holding plate is there. Here also there is a target cell. Inside there is a nucleus. The needle is loaded with the DNA molecule and also targeted exactly. So it will reach us the structure already we saw it. And after that, what happened? The structure size will be very large inside, otherwise called as what is the swellum of the pronucleases. Suppose if it is changed in any direction, this the holding plate is not properly hold the cell, means what happened? The target cell, it is not reaches exactly into the DNA molecule. Okay, that is very, very important. See the next one also already we saw it. And then liposome mediator transformation, liposomes. Okay, so that is also very, very important. Okay, but what happened, we are using some chemical substances that is called a spec. Okay, so it is similar to that of the liposomes. That is very, very important. Okay, so another one is called as ultrasonic, means already you know about ultrasonic sound. Ultrasonic sound means ultra violet laser microbeams also used for the uptaking of the DNA molecule. Not only that, and also the calcium chloride DNA precipitation. The calcium chloride is also used for the DNA precipitations. How it will be, it will easily penetrate the membrane permeability. It's allow the membrane permeability is more than that of the normal one. Okay, so already we saw it. This one, how much heat we have to give, how many times. The duration is also very, very important. 42 degrees Celsius up to 90 seconds only, 1.5 minutes only. Chemical agents, genes transfer. How? What are the chemical substances used for the otherwise called as agents, precipitating agents, destabilization agents? Okay, each one has a specific PEG, polyethylene glycol, and polyethyl ornithine, polyvinyl alcohol or divalent ions, that is called as calcium chloride. These are the chemical substances used for the gene transfer method. Okay, this is also very, very important, my dear student. That's all about, my dear student. In this part, we will see the next year and next part also. Thank you so much for the participation, all of you. Good luck, and I wish you all very happy New Year 2023. Okay, take care. Bye.
Thank you, sir.
Thank you. Bye. Thank you. Bye. Take care. Good luck.
Happy New Year, sir.
Happy New Year, my dear. Happy New Year, sir. Happy New Year. Happy New Year, Ajay.
Happy New Year, sir.
Happy New Year. Happy New Year, Ajay. Happy New Year. Convey to my wishes to your mom also. Okay. Thank you. Thank you so much.
Yes, sir. Thank you, sir.
Thank you, Kanna. Bye. Thank you so much for your guidance.